Let's transition to a look at the quantization of bacteria, and I want to think about the example that we have at hand. So this sample that we collected from the snow outside of the horse's pasture, which I have now taken and streaked onto a GSA, a TSA, and an EMB, so keep your hypotheses in mind about that. What if we wanted to count the number of bacteria that are actually in this vial? We literally wanted to count every single cell that was in there. How would we do it? Right now, you have a tool at your disposal that could allow you to do that. You could actually take a small aliquot out of that bottle, you could put it onto a microscope slide, and you could start counting. Sounds like fun, right? But that is one method. It's called a direct count. Literally, we directly count the bacteria on the microscope slide. This has some advantages because if I told you that it needed to be done by 2 p.m. this afternoon, you could get it done. However, it also has, as you might guess, a lot of disadvantages. Who really wants to count that number of bacteria on a slide? Not that much fun, right? But the other thing that's a big disadvantage of this method is that you really can't tell the difference between the living and the dead cells. So you're counting dead cells with wild abandon, and it's not giving you an accurate count of those cells in here that are actually living. That's why we tend to use a different method instead, right? Breathe a sigh of relief. We use the method that is called the standard plate count. So remember this idea that we learned early on, the ever important and epic concept that every single colony on an agar plate derives from one single parent cell in theory and evolves to an entire colony over the time of incubation. We're going to tap into that idea. So that means that if we take a sample from our vial and we put it onto a plate and we allow it to grow overnight, everywhere that a colony pops up, we would say that is one cell, that is derived from a single parent cell. So that's all good and well, but we have a possible problem. What if there are millions or even billions of bacteria in this vial? Then this is not going to work out so well, is it? We take this sample, put it onto our plate, and up grows a lawn of bacteria. You can't distinguish one colony from another because there's so dang many of them. That is where dilution comes into play. And that is going to be the concept of the rest of this part 2 vodcast. So in order to really get an accurate count of what's in here, it's likely that you're going to have to dilute it down into a series of bottles, and you're going to get to a point eventually that there's a low enough concentration in there that you can actually count it. So this is just a picture of what that kind of a dilution would look like, where you might take from your original culture and then put it into a bottle of water, dilute it a little bit, dilute it again, dilute it a third time, and then put it onto 3 different plates and see the average of the number of colonies that would grow up on those plates. So the method of standard plate count really requires you to first dilute the sample. So a liquid culture undergoes a series of dilutions. Set aliquots of the final dilute solution are spread on agar plates. Usually we take 100 microliters or 0.1 milliliters and put it onto the agar plate, spread it around with a hockey stick, and see what grows. Now, you're going to want to watch that on Virtual Edge. Let's take a minute to pull up that video so that everyone knows how to prep themselves to do this dilution. So here it is, Virtual Edge. You're going to want to go down to Lab 5 or Lecture 5 and find the experiments there. Experiment 12 is where we're going to be doing enumeration of bacteria, and you'll find the sweet epic pictures showing each individual colony that can be counted on a plate after dilution. And then, of course, you're also going to find the video that's going to show you how to do this process of diluting. So let's kind of get this queued up here so that we can kind of be watching what's going on in this video as the dilution takes place. So the set aliquots are going to be taken from the initial culture and then placed into the first bottle, and then of course diluted down from there. So here we're just seeing sterile technique. You're going to want to review this. You're going to want to go through this entire video and see the aliquot transfers from bottle to bottle to bottle. Now at the very end, you're going to be plating onto a plate and seeing overnight in an incubation what can grow on this plate. So you're going to spread it out with a hockey stick. It's pretty rad. So take time to watch that whole video and then understand that we'll be counting the cells, counting the colonies, which we know represent cells in the final aliquots. So be ready next time to get your Sharpie out and to tick off each colony as you count it. Then, of course, is when the math comes in. We're going to be working backwards from the first count that we get on the plates, working backwards to the initial culture to determine the titer of that original culture. Now, the sweet thing here is that that allows us to only count living organisms. We're not getting the count of the dead ones because they, of course, won't form a colony. But the hard part about this is that if you don't mix those bottles really well in between your transfers, it's possible that multiple cells could stick to one another, forming an aggregate. And then when that aggregate lands on the plate, it'll look like it's making only one colony. So then you have a false count because you're counting a colony that actually came from many parent cells instead of just one. So of course this method is going to require some math, and in a moment we're going to be going through that math step by step and figuring our way through a dilution problem so that we could take a final count on the plates and figure the original titer in the culture that you started from, or maybe the sample vial that you started from. Before I do that, I just want to mention that there are also indirect methods to do what we're doing. That is, sometimes you don't have, or maybe you repeat this experiment a lot and you don't have time to continually do the standard plate count. So people have found some indirect solutions to that. Rather than doing a direct count, they will use a spectrophotometer to measure the turbidity. Remember, we did something like that when we estimated the turbidity of our tubes. After the first lab, we mixed them and looked at how turbid they were, and based on that turbidity, we estimated the overall titer. Now remember, that's not a count, it's just an estimation. You can do that with a spectrophotometer to get a little more accuracy. You're still looking at the turbidity by looking at the optical density at 600 nanometers to see how much light it's absorbing. And you can have a calibration curve. You can actually make a curve that shows that for this OD, it is equivalent to this titer of cells. So this is something that's very time saving if you do this a lot. Likewise, you can measure the dry weight. So if you grew up a culture, spun it down in a centrifuge, and then dried the pellet, you can measure that dry weight and have, again, that correlation between dry weight is equivalent to about this titer of cells. Same thing with looking at metabolic byproducts. Maybe a culture makes some colored product, and you could measure the light absorbance of that product and equate that with the number of cells in the culture. So now let's jump right into doing the math, and I'm going to go through this in multiple methods so that you can be ready to rock and roll when you need to do your dilution problems. And actually, that's something you're going to be doing right away in class. Let's go ahead and take on a practice question. Here we can see that culture A was our beginning culture, and that it's been diluted by placing first a mil into 99, then 11 mils into 99, and 1 into 99 again, until finally the solution was dilute enough to plate it onto 3 plates, with each plate having 0.1 mils, or 100 microliters, spread onto the surface of the plate. Assume then that these plates were incubated overnight, and that on the first one there was 97 colonies, the second 100, and then finally the last 103 conveniently, so that when we look at an average, and the reason that we take an average here is to decrease random error, we see that we have an average of 100 colonies. But remember that that is representative of the number of parent cells that made those colonies. So on average, we have 100 cells that were found in every 0.1 mil aliquot. And eventually, remember that our goal is to find the titer of culture A. So we need to eventually express our our answer in a concentration, in the number of cells per milliliter. So we would want to do that here as well, noting that there was an average of 100 cells in every 0.1 milliliters. So at this point then, we can recognize that in bottle 3, the last bottle, there was concentration of bottle 3, an average of 1.0 times 10 to the third cells per mil. Now, by no means is that the concentration of culture A, but it is the concentration of bottle 3. And now that we have that concentration, we can very easily work backwards, first to bottle 2, and then finally to bottle 1, and then eventually in the end to culture A. So knowing the concentration of bottle 3 really enables us to do that. And you're going to have to remember an equation that maybe you thought you could forget from general chemistry. C1V1 equals C2V2. In this equation, the first C stands for the concentration over the more concentrated solution. So bottle 2 is going to have more bacteria in it, right, than bottle 3. So bottle 2 is the more concentrated solution. So in our case, this is the concentration of bottle 2 times the volume removed from bottle 2. So the volume removed. Now this is going to be equal to the concentration of the more dilute solution. So that's going to be the concentration of bottle 3. And then that's times the volume 2, which is the total volume of the more dilute solution. So V2 is, we'll say, volume total after the transfer of the more dilute solution. So let's practice plugging in for each one of these terms, beginning with CB2, knowing that it is our unknown. This is our actual unknown. We want to find out what is the concentration of bottle 2. So what is the volume removed? 1 milliliter. So let's plug in a mil for that variable. And then that is equal to the concentration of bottle 3, which we know, and we've already calculated as 1.0 times 10 to the third cells per mil. And then we have to multiply that by the total volume in bottle 3 after the transfer. So 99 plus 1, or 100 milliliters. Now I'm going to remove just a little bit of stuff here so that we can see what we're doing. So times 100 mils. Now this is what makes this method so fantastic, because we can cancel units. So we know that this is cells per mil, mils cancel, sweet. This is like my pastime in lonely nights. No, just kidding, but it is really awesome to see your units cancel, because it shows us that we're doing something right. Because at this point, when we divide through by the 1 mil, and we have 1.0 times 10 to the third times 100, well, that's 1.0 times 10 to the fifth divided by 1 mil. So the concentration of bottle 2 is equal to 1.0 times 10 to the fifth cells per mil. And there we maintain our units. That is a good looking concentration of bottle 2. So we've now worked back to the concentration of bottle 2. We're partway there. In fact, we're halfway there. We just need to go to the concentration of bottle 1 and then finally to the concentration of culture A. But in order to do that, I need to erase some of our messiness. We need to remember, though, that the concentration of bottle 2 is 1.0 times 10 to the fifth. So keep that in mind. Now that we have that written down, we can go back through that same calculation again. So we can recognize then that the concentration of bottle 1 times the volume removed, 11 milliliters, is equal to the concentration of bottle 2. We know that, 1.0 times 10 to the fifth cells per mil times the total volume, which we know is 99 plus 11, or 110. So times 110 milliliters. Unit canceling, Awesome. And we get 1.0 times 10 to the 5th times 110 divided by 11. Concentration of bottle 1 then is equal to 1.0 times 10 to the 6th cells per mil. Some of you just looked at that and said, wait, how'd she get there? Go ahead and just use your calculator, plug on in. Others of you are saying, well, yeah, of course, Rachel, that's a 10-fold dilution. Of course, it's going to go up by 1.0 times 10 to the 5th is going to go up to 1.0 times 10 to the 6th by a factor of 10. I don't care where you sit on that spectrum of math ability. It doesn't matter so long as you can get here either with or without your calculator. But we still need to do another calculation. We need to go to the concentration of culture A. So please bear in mind that 1.0 times 10 to the 6th is the concentration of bottle 1. We're going to erase and go to our last calculation. So in our final calculation, we're looking for the concentration of culture A. We know that that's times the volume removed, 1 milliliter. Then that is equal to the concentration of bottle 1. We know it's 1.0 times 10 to the 6th cells per mil. Now we take that times the total volume, 1 plus 99, or 100. We get jazzed as we cancel our units, and we go through the math. Concentration of culture A is equal to 1.0 times 10 to the 8th cells per mil. Wow. So look at the number of cells that were in that culture A. No wonder we couldn't just pour it onto the plate and proceed to count colonies, because of course there would have been no end and no beginning to all the colonies. It would have been a lawn of growth. But now, because of dilution, we were able to work backwards and know what was in that initial culture A. There's another way to do this. And some of you are thinking, gosh, I sure wish there was a different way to approach this problem. Well, there is. This is my favorite, however, because I love the idea of canceling units and seeing the units just fall into place. However, we can approach this in another way, and I'm going to show you that way now. This is a favorite amongst microbiologists because it enables them to do most of their math the night before their colonies grow. So what you do with this method is you actually identify visually every single location that a dilution takes place. You then go through and write for each one of those dilutions the volume transferred over the total volume, 1 over 99 plus 1, or 1 over 100. The same thing is true here, 11 over 99 plus 11, or 1 over 10. 1 over 99 plus 1, or 1 over 100. And then finally, here is our last dilution. Don't miss this one, 1/10 of a mil. 0.1 is 1/10 of a mil. So we now multiply all of these dilution factors by one another. I like to do that by putting everything into scientific notation. So 1.0 times 10 to the minus 2. Don't forget to check out the scientific notation tutorial in the back of your book if you are, has been a long time since you've done scientific notation. So then this is times 1 times 10 to the minus 1 times 1 times 10 to the minus 2 times 1 times 10 to the minus 1. When you multiply, you add exponents. So 2, 3, 4, 5, and 6. So this is a final dilution that is equal to 1 times 10 to the minus 6th. Now this is a dilution factor. It is unitless. But in order to understand what we need to do with it, I have an example. So say that you and your roommate like to share a pot of coffee every morning, but you like your coffee stronger than your roommate. So your roommate takes a little bit out and puts into her mug half coffee and half water. This we know then is half as concentrated as this. So if you wanted to figure out the concentration of the more concentrated coffee, you would multiply by 2, right? To get backwards to that initial solution. 2 is a reciprocal of 1/2. And that's what we need to do here as well. We're going to take the reciprocal of the final dilution, called the plating factor, which is 1 over 1 times 10 to the minus 6th. That is equal to, so the plating factor is equal to 1 times 10 to the 6th. That's what we're going to multiply by to get backwards to the concentration of culture A. But please take warning. 1 over 1 times 10 to the minus 6th is 1 times 10 to the 6th, because 1 over 1 is 1. However, be very, very careful at this step. Take note, because 1 over 2 times 10 to the minus 6th does not in any way, shape, or form equal 2 times 10 to the 6th. So be certain you're taking the reciprocal of this quantity and not just flipping the exponent. Wrong, 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 wrong. Don't do that. So now what we do with that plating factor is we take the average number of colonies, which remember has not changed. We still know that there is an average of 100 colonies, an average of 100 cells in that final aliquot. So we now just take that average times the plating factor. So to get the concentration of culture A this way, we get 1 times 10 to the 6th times 100, which is, yay, you guessed it, 1 times 10 to the 8th, the same answer that we got the other way. But now you have to add your units. So I'm going to do a little erasing so we're not too messy here. So 1 times 10 to the 8th cells per mil is the concentration of culture A. Cells per mil. Don't forget to add your units if you use this method. Answer. 2 ways to approach these problems, and I bet you can think of more ways. And I don't care which way you use, so long as you have a good approach to solving these problems. You're going to be solving these problems in lab today. And in fact, in the back of your book, in the appendix, you'll find the assignment that you're going to need to submit. Take the next few labs to get that done. Your TAs will tell you the due date, but please hold them to helping you in the labs and make sure you ask them lots and lots of questions. Hi everyone. So after a few days of incubation of the snow sample that I plated onto GSA, TSA, and EMB, our results are a little bit disappointing, but they're probably not surprising. So on the EMB, which we know to be both selective and differential, there really wasn't much in the way of growth. So we can see by taking a quick look at that there's just no real colony formation. Of course, that's not that surprising because it's selecting against everything that isn't gram-negative, and it's certainly also a less rich medium. So let's take a look too at, we know GSA to be a minimal bread and water diet, and we can see that the defined auger of GSA also did not support the growth of anything from our snowy horse pasture. But on the flip side, our TSA did. So we can see some pretty sweet single isolated colonies, not a lot of them, but these are going to be our examples of those bacteria that cannot synthesize their own growth factors, so they have to grow on this rich medium that provides preformed vitamins, amino acids, nucleotides, monosaccharides, right, building blocks for the synthesis of all of the macromolecules. So it's a good example of how TSA, the rich, undefined auger, supports more growth than the defined GSA and the likewise defined but also selective and differential EMB. Pretty cool.